Let me start with some introduction of the Mystery of ROS, which was held on May 11th this year. The conference consisted of 4 presentations: current status of increased ROS and diminished protective oxidative stress response, and key event component in LP development, presented by Yulia, and emphasizing the role of test methods in the AOP-Wiki by Clemens, and lessons learned from LP collaboration on COVID-19, presented by Carlie. This is a list of current ROS collaboration members. Approximately 70 people from 19 countries are listed here. These are important KEs related to the Mystery of ROS when it started in May 2021. And this is a Mystery of ROS collaboration network, and the report of the first and the second Mystery of ROS conference has been published in April 2022. And ROS-related LPs in LP-Wiki as of May 1st, 2023 are listed here. And oxidative stress-related LPs in the LP-Wiki as of May 1st, 2023 are listed here. And these are ROS-related KEs in the LP-Wiki as of May 1st, 2023. And these are ROS oxidative stress-related KEs in the AOP-Wiki as of May 1st, 2023. These KEs are more closely introduced, and I would like to focus on introducing KE 1940, increases in several reactive oxygen species, and KE 1869, diminished oxidative stress response in the development of AOP-298 and AOP-379. I would like to introduce development of AOP-379, binding to S2 leading to thrombosis and disseminated intravascular coagulation in International Joint Research Project CHOW, which was led by Carlie. And microarray data and RNA sequencing data on the database were analyzed in terms of gene expression profiling, and molecular pathway network analysis was performed using Ingenuity Pathway Analysis (IPA). In 2020, we developed AOP-379 as increased susceptibility to viral entry and coronavirus production, leading to thrombosis and disseminated intravascular coagulation. In order to elucidate the molecular networks related to the coronavirus infection, gene expression data of cells infected with severe acute respiratory syndrome coronavirus 2, SARS-CoV coronavirus 2, on the coronavirus pathogenesis pathway were analyzed. Literature on coronavirus has been investigated to reveal the mechanism of COVID-19. The relationship in ROS, coagulation, and SARS-CoV-2 have been reported. And the relationship in ROS and RF2-mediated oxidative stress response in SARS-CoV-2 has been investigated. The relationship in ROS and RF2-mediated oxidative stress response in SARS-CoV-2 has been investigated. And we discussed on CHOW LP379 network several times in CHOW meetings. And the reactive oxygen species, ROS, play crucial roles in a variety of diseases and physiological conditions. ROS are produced by various stressors such as chemicals, particles or radiation energy, and endogenous peptides on the enzymatic machinery. The international online conferences on the mystery of ROS took place on May 31st, 2021 and October 8th, 2022. At the first conference of Mystery of ROS 1, a brief introduction of the ROS collaboration was followed by 8 presentations. The Mystery of ROS Consortium carefully considered many critical aspects of the creation of ROS-related KEs. It was identified that ROS are needed for critical cellular functions at optimal levels. However, excessively high levels of ROS production over prolonged durations lead to adverse effects. Thus, the cellular balance between ROS production and scavenging to maintain homeostasis needs to be represented within the case. The second conference on the mystery of ROS included 4 presentations and panel discussions on DNA damage and ROS on nanomaterial-related oxidative stress. The initial concept for ROS-related KE group was introduced, which led to the creation of harmonized KE9040 upregulation of ROS, currently called increase in cellular ROS, in collaborative effort of conference attendees as in the figure. Hexagram star shapes indicate the ROS-related KEs. Pentagon shapes indicate DNA damage-related KE. Dodecagon shapes indicate coronavirus-related KEs. KE1940 focuses on the production of ROS per se, while the reduction of protective enzyme activity that also results in ROS increase will be the focus of the KE1869, depletion of or diminished protective oxidative stress response. At molecular and cellular level, KEs for ROS. The review of the mystery of ROS consortium has been published in Frontiers in Toxicology in 2022. And the diagram of AOP-379 has been changed as in the slide. Current AOP-379 consists of MIE 1739 binding to ACE2, K1738 SARS-CoV-2 cell entry, and K1901 interferon-1 antivirus response antagonized by SARS-CoV-2. K1847 increased SARS-CoV-2 production, and K1869 diminished protective oxidative stress response, and K1845 coagulation, and AO as K1846 thrombosis and disseminated intravascular coagulation. So these AOPs 298 and 379 are currently developed in OECD. So these are ROS-related KEs proposed as umbrella KEs. K1940 increases in cellular ROS can be umbrella KEs for all KEs related to ROS increases. And K1869 diminished protective oxidative stress response can be specific KE focusing on decrease in protective oxidative stress response. And K1392 oxidative stress can be an umbrella K for K9040 and K1869. So molecules related to ROS production, oxidative stress responses, and blood coagulation were mapped on the coronavirus pathogenesis pathway. Literature studies have suggested that ROS, oxidative stress response, blood coagulation, and thrombosis are involved in COVID-19. ROS-related KEs have been harmonized in an international consortium, Mystery of ROS. So I would like to introduce the concept of umbrella or representative KE, which was presented at Handbook Guidance meeting in 2021. Nowadays, some similar KEs can be found in the AOP-Wiki, as developing new AOPs and recycling existing KEs. An umbrella KE may be defined as a concurrence of set of similar KEs. Let's say there are kind of similar KEs, such as club 3, 6, and 9. And in case you cannot choose one of the club KEs, this big club KE, which is so-called umbrella KE, may be substituted for the all synonym KEs. Since the existing KEs such as hard KE, alpha, beta, and gamma have been developed in accordance with the existing AOPs, it's sometimes too specific for a new AOP you would like to start to develop. And this is just an equation in which I thought that the concept of umbrella KE is represented. So umbrella KE or KE hat equal to KE alpha cap KE beta cap KE gamma. So umbrella KE represents a set of similar KEs, which I hope is categorized in ontology base or terminology base. These are pictures of Plunam mume, Japanese apricot ume, which includes various kinds of Japanese apricot varieties such as Chugoku no ume, Fujibotan, Renkyu, Benikaga, Shirokaga, etc., etc. So it is said that more than 500 varieties exist in Plunam mume. Then this mon in Japanese language, or icon, or pictogram, or pictograph, or symbol mark corresponds to umbrella KE in this concept. And in case someone is developing a brand new AOP, you may look for existing AOPs as AOP alpha or AOP beta. So for example, for AOP alpha, this Benikaga red ume KE is so important and it cannot be changed. And Renkyu and Shirokaga white ume KEs are very important for AOP beta as well, and they cannot be modified for a new this Humpty Dumpty AOP. So in that case, the umbrella KE plus mume or ume representing all these three KEs can be selected for the new AOP Humpty Dumpty leading to amygdalas, Persica Jean-Baptiste Chevalier de la Mare, peach, momo in Japanese language. So in summary, there are needs in umbrella or representative cases. So in this case 1, people on the earth says this is an UFO, unidentified flying object, or UAP, unidentified aerial phenomena. On the other hand, people from the galaxy says this is our plane, not UFO. So we need some umbrella or representative name for that. And in the case 2, people from the galaxy and people on the earth have many types of flowers in different shapes and in different colors, and so many types of flowers are here. And tulip, rose, cherry blossom, plum, lotus. So they are different flowers, but they are all flower. So they might be in red, in blue, or they might be big flowers or small flower. But we need some kind of umbrella or representative flower for summarizing or to indicate the flower characteristics. So we need a representative flower shape which have common characteristics or features as beautiful. So I would like to acknowledge all the collaborators and people, and thank you very much for your kind attention.
Thank you so much, Shihori.
Thank you.
Did we have any quick clarifying questions? Again, the goal is to save the discussions for the breakouts. So we'll try to keep the questions in this session just to short clarifying questions. Is there any quick questions for Shihori? Okay. Well, we'll look forward to tackling this topic in more detail in the breakouts. Thank you, Shihori. And I believe the next speaker is Carlie LaLone.
All right. Are you viewing my screen?
Yes, I can see.
Great. So I'll be talking about capturing domain of applicability in the adverse outcome pathway wiki. Advance my slides. So just as a reminder of the current domain of applicability in the AOP-Wiki for the overall description of AOPs, as well as key event and key event relationships, there's these various categories such as life stage, taxonomic applicability, as well as sex applicability. And with each of those, the developer is able to capture information on the level of the evidence, as well as provide some free text to provide like the input for, manuscripts, for example, or where that information is coming from for the taxonomic domain of applicability, sex applicability, and life stage applicability. So to move forward, some of the objectives would be to be able to capture the evidence to understand domain of applicability, as well as thinking about the biologically plausible taxonomic domain of applicability and being able to pull that into the key event and key event relationship tables. And, thinking about conservation and thinking about the domains of applicability, we're really looking about structural and functional conservation. So we needed ways to be able to guide specifically how you could incorporate domain of applicability in key event and key event relationships, particularly the challenges that within the AOP-Wiki, many times there's very limited information, empirical information specifically to help define the TDOA. So we might get, a species relevant to an AOP or a key event relationships, or a handful of species, just based on the availability of data to support the development of those elements of the AOP. And so there's opportunities in advances from some of the computational approaches to define the biologically plausible taxonomic domain of applicability. And I'll be speaking about that, centered around some bioinformatics approaches that can bring in greater numbers of species and taxonomic groups to the conversation, and thinking about specifically in the taxonomic domain of applicability. So there could be some guidance for how to bring in various in silico, in vitro, and in vivo data to provide the weight of evidence for the biologically plausible TDOA. And then, there's also an importance when we're thinking about domain of applicability and the adverse outcome pathway framework and the use of the adverse outcome pathway framework in decision making. So understanding, where there is conservation, but also define where there is a lack of conservation as well. So that may be something for consideration, because that type of data is relatively readily available in many instances. So I also wanted to then bring together a discussion about the AOP-Wiki to accommodate the data connectivity between the domains, between the life stage, sex, and taxonomic domain of applicability. So just speaking about the taxonomic domain of applicability, I wanted to just define two pieces of information, because I've been saying the biologically plausible taxonomic domain of applicability. So in our terminology of the current AOP-Wiki, we talk about domain of applicability, and that brings in information from empirical data that we have supporting whether or not a taxonomic group or specific species, whether or not that molecular, or, the key event relationship is relevant for that group of species or taxa. And that typically comes from in vitro and in vivo evidence. And that, of course, would come from a handful of model organisms where there is data that exists. But there's also the ability, like I mentioned, to bring in bioinformatics data, which allows us to give predictions based on evolutionary relationships, based on understanding of conservation of genes, proteins, for example, to be able to predict from the empirical evidence that we have hundreds to thousands of species for which the information would be relevant for in defining taxonomic domain of applicability. So just as an example of this, my group has published on defining the biologically plausible taxonomic domain of applicability for an adverse outcome pathway, and laying out the process for being able to utilize bioinformatics approaches and incorporate those into the AOP-Wiki. And so that information has been integrated into the AOP-Wiki handbook currently as far as how you could go about defining the domain of applicability, utilizing not just the available empirical data, but also available bioinformatics approaches that allow us to define biologically plausible as providing scientific evidence through understanding conservation of various elements of the adverse outcome pathway. And I'll chat a little bit about that, what's in the handbook. So this is a busy slide. I'm going to try and break it down just a little bit. We're all used to seeing the box and arrows with the molecular initiating event, key event relationships, key events, etc. So that's what I have just up top to kind of orient everyone. And then typically what we're used to seeing as well is the in vitro and or in vivo evidence for that particular element of the AOP. And we're talking about defining the taxonomic domain of applicability. That information that's captured is, it may be something as broad as teleost fish, the scientific term. A lot of times this is a species common name, a species scientific name, the evidence call, and then a link to NCBI for each element of the AOP, whether it's a key event relationship. So I define that here. So you have your empirical data that defines the taxonomic domain of applicability for each key event and key event relationship as you go down the AOP. So what the proposal would be is that we also allow for the integration of bioinformatics approaches for species extrapolation that take advantage of what we know from the empirical data. So say that we have understanding of the molecular initiating event as far as, the gene, the protein. There's methods that exist to allow us to understand how well conserved that biology is and provide information and specifically lists of species to be able to expand the taxonomic domain of applicability, like I said, from the single species to perhaps hundreds of species, thousands of species for which, we can't test empirically, or we don't have and won't have empirical data, though it would be important from a regulatory standpoint, thinking about, just as an example, endangered species, and in thinking about, if you're using the tool to understand a specific impact on various species within, your region or your global entity, regulatory entity, you may be interested in particular species, but that information, doesn't necessarily exist because of the lack of empirical data. It's derived in vitro or in vivo. So there's in silico. And here I use an example of the sequence alignment to predict across species susceptibility tool, but there's certainly others that can provide this type of information as well. And so therefore you're expanding from a handful of species, defining taxonomic domain of applicability to a much broader diversity of species. So again, the empirical data would define the taxonomic domain of applicability, but we can use bioinformatics as a means to define the biologically plausible taxonomic domain of applicability. So this is what is presented both in our Jensen et al. Paper in 2023 here that defined how you would go about using bioinformatics to understand taxonomic domain of applicability. And basically, it's just exactly what I showed, but in a different format where you're looking at the empirical data that exists to define each taxonomic domain of applicability for the key events. And then once you've defined that, you can take that biological evidence and generate biologically plausible information from the bioinformatics approaches. So you get another list of species for each key event that has empirical evidence, bioinformatics evidence, and then you can take the upstream key event to the downstream key event in those lists of species or taxonomic groups, and you look for the narrowest common species between the upstream and downstream key event to be able to define the taxonomic domain of applicability for the key event relationship. And then further, kind of following along with that same model, because we define the key event TDOA, the key event relationship TDOA, and then the overall AOP TDOA, we can then take information from the biologically plausible taxonomic domain of applicability for the key event relationships throughout the AOP and pull those together, again, looking for the common species between the key event relationships to define the overall AOP, taxonomic domain of applicability. And again, this is presented in a publication, but also in the user handbook for the AOP-Wiki. So then, so we're expanding the information that's available for users to be able to understand TDOA across the diversity of species, but then further, there would be a desire to be able to integrate TDOA with the other domain of applicability considerations, as well as the weight of evidence. So how we could go about doing that. So this figure up on the right is just to kind of remember and orient us with what we just chatted about as far as the upstream and downstream key events and merging the species lists for defining taxonomic domain of applicability. So you can see that it would continue on with the same sort of format that we have currently in the wiki, having common name, scientific name, as well as evidence calls for those particular species and links out to the NCBI, or the National Center for Biotechnology Information Databases, to be able to link to information there. But then you would also be able to integrate within that same table, life stage, as well as the evidence for the life stage, as well as sex and evidence for sex for the upstream and downstream key events. So it kind of brings together the full picture for domain of applicability. Instead of individual tables, it has it all, together, because sometimes you can see certainly differences in the different sexes and in life stages, for example, among species. And I think that also is where it's more detailed and it's more information for the user to be able to apply these approaches for decision making. And then taking that a bit further, excuse me, you could expand upon the data for which exists for defining that weight of evidence. So for example, like I had mentioned, with the taxonomic domain of applicability, you're capturing in silico data. Now in silico derived predictions of susceptibility would not give you the highest evidence call, right? So it's a prediction. So you would consider that as the computationally derived predictions of taxonomic domain of applicability, the biologically plausible TDOA, as a low evidence call. However, when you can partner that with in vitro and in vivo data, you can move that to high call of evidence for taxonomic domain of applicability. So basically, you capture where that data is by entering that information of where the source data comes from. It provides you with the evidence call, which there could be links out from the evidence calls that provide you with that free text that captures where that information came from. If it is an in silico approach, it could describe the approach and the case studies that were developed to support that particular call of conservation that would lead to support in this particular table, as well as the in vitro and in vivo data, which is already what is captured in the wiki currently, but it gives more information for the use and the ability to use third-party generated data from bioinformatics approaches to support evidence as well. And so you can see that there'd be the links out to those free text pieces of information. Then the other question comes in of whether or not we should consider evidence for both conservation of the biology as well as lack of conservation. And the reason I put this in there is because this data is readily available and generated from some bioinformatics approaches, including the CCAPAS tool, which my team develops. So just continuing on with integration of the TDOA with the other domain of applicability, considering evidence against conservation, you could envision that there would be, for example, data that also you could provide the high, moderate, and low calls for example, if invertebrates are not involved in that particular piece of biology, you could provide in silico, in vitro, and vivo support to capture that knowledge as well, which may be, in speaking with some of our program office partners, just as important in decision making as knowing what species and which taxonomic groups are likely to have that biology conserved as well. So there's opportunities there by integrating bioinformatics, not just to define the TDOA as far as those species likely to maintain that biology, but also those that are less likely or unlikely to maintain that biology as well. And then this was meant to just be a slide for if people had questions, but basically an example using the CCAPAS tool that provides information on conservation across the diversity of species by looking at protein sequence and structural conservation. And basically, you know, the output from a CCAPAS evaluation provides the information that's already integrated in the AOP-Wiki as far as the NCBI accession, the species, both the common name and the scientific name, and whether or not it would be likely conserved or unlikely conserved. So just as an example, this type of an approach that I described, CCAPAS and the data that's developed there is readily able to be input into a format for which I was describing for some of the in silico pieces that could define the biologically plausible taxonomic domain of applicability. And I think this ends up solving some challenges for which we don't have a great deal of information filled out in the AOP-Wiki relative to domain of applicability, and particularly thinking about taxonomic domain of applicability as well. So those are ideas for consideration in the discussions moving forward.
Thanks, Carlie. Are there any clarifying questions? Well, this was great. This is definitely going to set up a really nice discussion when we move to the breakouts. So I guess I'll hand it off to Dan.
All right, thanks, Steve. So the last topic today was just discussing this idea or question that was raised in one of the earlier sessions of this workshop. This question about the AOP-Wiki as a registry. So the question sort of came up, what is the AOP-Wiki really intended to be? Is it a traditional wiki in the sense that it's a creative space for exploration and flexibility? It's designed to be populated by the crowd, whatever the crowd is, and that curation and evaluation of the information just comes from that crowd, and we rely on the crowd to verify and, you know, assess the validity of the information that's contained, versus the concept of the AOP-Wiki as a registry that is a collection of official records that are, say, endorsed or overseen by some authoritative body that vets the information that's there, makes sure it's of appropriate technical quality and content, and can really be used to support decision making and isn't full of errors and so on. And, you know, we see the same discussion around content that's in Wikipedia, for example, where, you know, a nefarious actor or whatever can go in and change pages and, you know, put in some completely fabricated information, for example, but we rely on other users in the crowd to correct that, to point out where things are wrong and to restore the original information, versus something where all the information is vetted ahead of time and you know going there that anything you see there is reliable and good to use. So this concept of, you know, sort of what is the wiki, what should it be in terms of this level of information, kind of ties with a tension, if you will, or a tug of war between kind of how we develop the wiki. So on one hand, you have needs of the content developers or the AOP developers in terms of how they want to enter the information, flexibilities they want, keeping the burden low, making sure they get some kind of recognition for what they're putting into the AOP-Wiki, and the natural tendency for scientists to want to, assemble a lot of evidence and address a lot of the different issues and angles and nuances in their data versus the desires of the content consumers to make sure that information is very systematically organized, it's vetted, it has gone through levels of scrutiny that maybe place a lot of burden on the developer, and that is very scientifically robust and supported, yet simple to understand, short and concise, etc. And so trying to find the right balance between these two things is a difficult challenge, one that we've struggled with in the wiki from its inception. And in general, I'd say we probably have weighted more heavily toward the needs and desires of the content developers, simply because those have been the folks in the room as we've talked about different features of the AOP-Wiki. So, so again, you sort of have these different needs where you have this common knowledge base of information that basically documents, stores the AOPs, disseminates those. You've got content developers that are putting information into this system, and you've got the content consumers who are extracting information out of that system for a given application, and their needs are slightly different. So for example, the content developer really may want the ability to crowdsource and distribute the labor of developing AOPs, build on the expertise and the contributions of others, the ability to collaborate, avoiding doing something that somebody else has already done, being able to clearly see where there's gaps, and also leverage the emergent knowledge that can come from creating links. But on the other hand, you have content consumer who needs a system that's very accessible, searchable, systematically organized, has everything transparent and well documented in terms of all the supporting materials. Ultimately, needs to know that what they're seeing is scientifically defensible and credible, and that it's presented in a form that's simplified, synthesized, and translated. So the needs may be somewhat different. Back in 2018, the AOP Knowledge Base Working Group at the time kind of talked about this idea of whether there should be separate systems. We talk about the AOP knowledge base as really this database full of the information, and that the interface does not necessarily need to be the same for content developer and the content consumer. And right now, that's really what the AOP-Wiki itself is. It's both the developer's interface, how information is populated into this knowledge base, as well as the view that people get when they're pulling information out. Now, of course, third-party applications can also access this information and display that in potentially different ways. And so this sort of vision that was laid out in 2018, I don't think we ever necessarily discussed this in a formal way, but this really became sort of the model that we see today in terms of how the AOP knowledge base is presented. So this is kind of that central storage place for all the information, the various tools that interact with that knowledge base, and the fact that the AOP-Wiki is really the only tool at present that allows you to add information to the knowledge base. The rest of these really are pulling information out. So the question is, if we have this kind of structure in place, and if we think about the AOP-Wiki as the authoring tool, is there room for developing new sorts of interfaces that the content consumer would be tailored to their specific needs. So for example, if you're a regulator and you need and want only the well-vetted AOPs, only those that have been endorsed, registered, if you will, have all the supporting documentation organized in a way that makes it useful for you, is there another interface or dashboard, if you will, that provides the information and filters out all the things that are maybe under development, lack sufficient development, etc. Whereas if you're, say, a research scientist and you're going to the AOP-Wiki with the intent of saying, what do we understand about this particular biological pathway? And what are the knowledge gaps that maybe I can focus my research on in order to improve our understanding of this biology or toxicology? Maybe that interface needs to look different. And so what are the characteristics of how that might differ and what kind of AOPs you would want to be able to view versus filter out, etc. And the same goes for, say, a computer scientist or a modeler who is interested in computationally accessing the information, making sure that information is machine readable, etc. So I don't have any solutions. This is really just intended as a thought starter. But we can conceptually see how you could build these different interfaces or dashboards to interact with or access the information from the AOP knowledge base. But some of the challenges that come with trying to develop what this content consumer interface should look like is really getting the stakeholder input to really understand and know what do these different consumer bases want, and then providing the resources and the time to actually develop those custom sort of interfaces. So with that, I will stop sharing and tee that up for discussion.
Any clarifying questions for Dan? If not, so we had a break schedule. Hi, everyone. Welcome back. So the consensus seemed to be that we just do a short recap of the three groups. And maybe we can start with the umbrella key event group, since you had the most people and have said that you've solved all of the issues.
Yeah, sure. Thank you. I was somehow nominated as the moderator there, and so we added lots of stuff to the mural thing, and let me briefly go through that. So umbrella key events, I mean, we identified that there is an overlap with gardening activities. So meaning that only in a well-gardened wiki, you would start putting stuff under umbrella key events. We said that there would be a new page type. So an umbrella key event page type would be slightly different from the normal key event page type. And the low-tech solution for summarizing certain key events under an umbrella key event would be tagging, the tagging solution that has been already mentioned in our first meeting on 26th of July, meaning that you would have a tag to which in each key event page, to which umbrella key event does this key event belong, and the authors would be free to just add a tag for an umbrella key event who someone would have to have created. So the umbrella key event page would have someone else has already created that. The umbrella key event page would contain some guidance, some information to AOP authors, telling them if you think that your key event fulfills this and this and this prerequisite, then feel free to add it to this umbrella key event. In terms of visualization, we think that a box inside a box, boxes inside of bigger boxes, and the bigger boxes being the umbrella key event would be nice. Maybe in a different shape, not the box, but like we have in our mural here, a pentagram here would be, or some other, not the box, but not the square, but some other geometric shape. So what else did we say? Having people voluntarily add their key events to an umbrella key event would also help the gardeners to find double entries. So let's say someone doesn't really feel like he would add his information to another key event, but would prefer to put his new key event under an umbrella key event. And the gardeners then find out now, well, actually, these two key events are the same, then they would much quicker identify such situations. What else do we have? So we have identified that this whole thing would be an end-to-end relationship, meaning one umbrella key event can obviously have several member key events, and one key event can be part of several umbrella key events. We have not yet decided on what is actually the best word for member key events. Is it child key event? Is it member key event? So we have not yet agreed on that. Then we also said that there should be no nesting of umbrella key events. So an umbrella key event should not be able to become member of yet another umbrella key event. So no nesting. What else? So yeah, the main thing is the umbrella key events would not necessarily absorb the member key events, but keep them as separate key events, because that was one of the first, let's say, decisions we took. No, umbrella key events would not suck up the member key events, but they would leave them as separate key events. And then the tagging idea came up. Yeah, I think, I mean, if you want to get an overview of all our discussion points, just have a look at the mural board in our section. But I stop here. People from our group, do you have anything I forgot? Do you want to add anything? Okay, if that is not the case, then thank you, Shihori, for raising the issue, and thank you everyone in the group. And I hand it back to Steve.
Carlie, I guess we had the next largest group. Do you want me to try and summarize? Do you want to try and summarize?
Yeah, I can summarize.
Okay.
Yeah. So I guess, you know, we chatted about the assumption of applicability. We chatted about, you know, whether or not the assumption is that unless there's evidence that suggests otherwise, we assume that there's taxonomic domain of applicability, or you know, we talked about capturing the evidence against, or the evidence for a lack of conservation, and the importance of having that information for a variety of different stakeholders. We chatted about better being able to tag some of the TDOA information for searching and browsing, and how we may be able to leverage existing, you know, taxonomic lineage from, say, NCBI or, you know, others, but also recognizing that different regulatory bodies utilize, you know, different groupings. We talked about combining the domains of applicability, and how there are efforts underway that would support the ability to be able to combine TDOA, as well as there are efforts underway that would be useful for capturing, you know, the in vitro and in silico, or excuse me, the in vitro and in vivo data as a table, and then how we could integrate in silico data as another piece of evidence as well, for the user to be able to make a determination on the weight of evidence. We talked about third-party tools such as, for example, CCAPAS and their ability to be able to be developed so that output would readily be integrated, at least from an in silico side, into the table formats or whatever formats are defined for domain of applicability, so that tools could readily be integrate their data. Let's see. We also talked about how it becomes important in instances where there, may not be empirical evidence. So, for example, if a key event, there isn't a defined taxonomic domain of applicability, as an example, what do we do in those instances for defining the overall AOP? And so we did talk about other types of extrapolation approaches and thinking about biological plausibility and the use of traits from the species that we do have information, and how that could be incorporated as well to be able to understand taxonomic domain of applicability and other domains of applicability. So beyond kind of the slides and what we presented, those are some of the expansion topics that were discussed during the breakout. Did I miss anything from those of you that were in that breakout group?
I think you captured it. I think, we did discuss that, one of the bigger challenges is going to be thinking about, because we've kind of always thought in terms of empirical evidence for, and so the bigger challenge is now thinking about, okay, how do we represent evidence against, and how do we incorporate the bioinformatics and potentially the diversity of different lines of evidence that could inform the biological plausibility. So and that kind of bled over into, which is a nice segue to Dan's topic, the different wikis for different users, right? And is there, as we start synthesizing the information, does it, for one purpose, would people want it one way, and for a different purpose, would they potentially want it a different way? So maybe with that, we can hand over to Dan to close us out.
Sure. We didn't have a lot of discussion on that particular topic in the breakout. So it was just you, Song, and I, and we had a chat about a wide range of things related to the wiki, everything from how we can capture the evidence that goes into the KERs as tables and be able to export that evidence, which ties in with what Jason presented on kind of the evidence mapping application potentially for the wiki. Talked a bit about endorsed AOPs, how those are being used, and whether they're treated differently or not. And I think that kind of ties with this issue of different interfaces for different users and the ability to filter. Many regulators only want to see the endorsed AOPs. So, that's some of that serving up what people want, the ability to filter on those attributes of the AOP. We talked a bit about how to capture quantitative understanding and evidence in the AOP, and whether that would be part of the evidence entry into the AOP knowledge base. And I think some of Jason's early mock-ups had fields where you could actually enter dose response kind of information or time course information, that sort of thing. But we want to be cautious not to make that a required field, because that puts a lot of extra burden on the developers that they may not want to do. Talked about some of the challenges of the difficulty in connecting in vitro to in vivo types of data when building those quantitative relationships. So a lot of times they're not immediately translatable between things that you can measure in an in vitro system versus the effects that you might measure in vivo. So how do we bridge that gap in terms of how the quantitative information can be captured? Talked about communication within the AOP-Wiki and how sometimes reaching out to authors of a key event or key event relationship, you don't get a response. So it's not clear whether you can go in and edit their page or not. So we talked about how some of the data model and the contributor tagging might help support that. And I think that's kind of a listing of some of the things we chatted about. So, yep.
Excellent. Well, we've done a great job. We're right on time. So anyone have any closing thoughts?
I guess I had one question maybe related to the umbrellas, and I wasn't involved in the discussion, but I was just curious whether an umbrella is fundamentally different from a tag or just a listing, like a group of key events that share some kind of connection or relationship. And it relates a little bit to the taxonomic things that Carlie and company were talking about in terms of like, could you create an umbrella for key events that are relevant to fish, for example? Or is an umbrella only something that pertains to a specific biological event? It's essentially like a more generalized form of a key event, or can it be other types of groupings, taxonomic or developmental, or all the key events that can be measured with an ELISA? I mean, I was just curious whether there was any discussion about that or views on that.
Well, maybe the umbrella KE maybe generalized KE to include many specific KEs in the same category or in the same ontology or similar ontology. But I think it depends on your thoughts or developer's thoughts. So there are many options for umbrella KE concept, but yeah, my understanding would be the generalized KE to
Okay.
many cases.
Yeah, we did discuss indeed this briefly. I would agree with what Shihori said. So any of the scenarios that you outlined then would be possible use cases of umbrella key events. And so there would be more than just tagging of KEs belonging together as a group. For example, we discussed that an umbrella key event should also have the option of having its own DDoA description, for example, which is something that in the example that we presented in the previous session on the thyroid network, AOP network, is something that we would require. So there we would have the example where you would have, for example, increased T3 and increased T4, and so all of the thyroid hormones grouped as one umbrella key event, and that umbrella key event in and of itself would have its own TDOA description that we would actually use as a way to describe the overall TDOA of the network. So basically, what of the properties and features that you would fill out or would not fill out for an umbrella key event compared to a regular key event would basically depend on the reason why you create the umbrella key event and what you want to do it, what you want to use it for, and what the application is that you have in mind for creating the umbrella key event.
Yeah, interesting. So I don't want to blow open a whole new discussion here, but yeah, really interesting.
Yeah, and tagging was mentioned as a low-tech solution for implementing the umbrella key event. So you would have a separate page for the umbrella key event describing the reason why it exists and what qualifies a member key event to become part of that. But then the actual implementation would be just a tag in the member key event, a tag leading to this umbrella key event. And the reason for creating, as my colleagues just said, the reason for creating an umbrella key event can be all kinds of things. Similarity, what you just said, yeah. So.
Okay. Well, I think we've reached the end of our time. Hopefully everyone can join again in, what is it, 5 hours, I guess, from now, for session 2B, where we'll have 3 more topics, and look forward to continuing the discussion.
Okay, see you all again.
Thanks, everybody.
And Dan, just to confirm, so you're running session 2B, right?
That's what I've been told. So,
All right, very good.
all right. See ya.
Bye.
Bye, everyone.
Bye. Thank you.